Dear students, in this lecture we will discuss about the functions of metalloproteins. We had already discussed the heme proteins and now we will discuss about the metalloproteins. Metalloproteins may include heme proteins also. So heme proteins can be under the category of metalloproteins. In heme proteins, the active site is iron actually, and in metalloproteins, the active center may be any kind of metal center. So metal plus protein, they are together known as metalloproteins. Proteins are actually the main constituents of the biological systems, and these biological systems they catalyze a number of biochemical reactions. Okay, and these amino acids they are formed of amino acids, and these amino acids actually they are bind with some metal ions, or they may be bind with the clusters, or they may be bind with the molecules as their active sites. So the active sites of these amino acids sequences that are the proteins, they may be the metal ions, they may be the clusters, or they may be the organic molecules. And generally, metal ions are the active sites of the most of the proteins that are used for their enzymatic actions. So, in the enzymes, various enzymes or various metalloproteins, metal ions are the active sites. So, we are taking the examples of the proteins that contain metal ions as their active sites. These proteins are called as metalloproteins because the proteins they have the metal center as their active site. And the metal center that is present in the active site of the metalloprotein, it is responsible for various functions of these proteins. So these proteins are active due to the presence of the metal center in their active sites. Like this iron, manganese, copper and molybdenum, they are the active sites of some metalloproteins and that are responsible for oxidation and reduction processes. Likewise, in case of cobalt, if cobalt is the active site of any protein, it causes radical based rearrangement and methyl group transfer reactions in the biological systems. And if the active metal sites are zinc, iron, manganese, then magnesium, nickel, they can be utilized in the hydrolysis processes. Likewise, the zinc in DNA processing and various kinds of metalloenzymes, they are involved in the storage and transport of various proteins, enzyme, signal transport proteins. And another examples of the metal containing proteins, they can transfer the electrons, they can transfer the oxygen and carbon dioxide, they can transfer the uh, electrons, they can transfer the energy. So there are various functions done by these metal centers when they are present in the proteins. Now, if we see the coordination around the metal center, so what are the atoms or groups they are present around the metal center as their coordination, then we usually found that the nitrogen atom, the oxygen, the sulfur atoms that are present in various amino acid residue, they act as the coordinating sites to the metal centers of the metalloproteins. Okay, so like imidazole group of histidine. So imidazole group of histidine, it contains nitrogen. Thiolate group of cystinyl, that is sulfur present in the cysteine group, and carboxylate group of aspartate are the main functional group that are directly attached to the metal centers of the metalloprotein. So generally nitrogen, oxygen and sulfur are the donor atoms that are present in the coordinated ligands to the metal center. Sometimes the macrocyclic ligands like porphyrin ring, they also present around the metal ions of the metalloporphyrin. Again, these macrocyclic ligands, they have generally nitrogen as their coordination site. Now we will discuss one by one the different functions or role of metalloproteins in the biological system. So first we will discuss about the role of metalloproteins in electron transfer. What kind of metalloproteins they are helpful in electron transfer? So main three metalloproteins, one is cytochrome, the second is rubridoxin, and the third is the plastocyanin. These three are mainly responsible for electron transfer, and these are the metalloproteins. So first we will discuss the cytochromes. So these cytochromes, they are actually the membrane bind heme proteins. So why they are heme proteins? They are heme proteins because they have heme as the prosthetic group, and in heme they have Fe, that iron in 2 plus form, and cytochrome have iron in the heme group, but the substituent of the porphyrin ring are different. So there may be three kind of cytochromes: cytochrome B, cytochrome C, cytochrome A. So cytochrome A has the heme as the prosthetic, heme A as the prosthetic group, cytochrome B has the heme B as the prosthetic group, and cytochrome C as the heme C as the prosthetic group. And electron transfer takes place like this: from cytochrome B to cytochrome C, and from cytochrome C to cytochrome A. So this is the electron transport chain. And cytochromes, they also helps in the generation of ATP through electron transport. So when the electron transport takes place, the production of ATP will also takes place. And during this electron transfer, the iron center will the iron center will oxidize or reduce during this electron transfer process. So this used the redox behavior of cytochrome P450 is used as an enzyme in the biological systems, and it will be discussed later on in the role of metalloproteins as enzymes. So these three, cytochrome B, cytochrome C, and cytochrome A, they are they perform the function of electron transfer in the biological system. Rubredoxin is another kind of metalloprotein that involved in the transfer of electrons in the biological system. So if we see the class of these kind of metalloproteins, so these are low molecular weight iron containing proteins that are present in sulfur metabolizing bacteria and archaea. So these rubredoxins, they are not present every in each in this biological system, but they are found in the sulfur metabolizing bacteria and archaea. In contrast to the iron sulfur proteins, rubridoxin do not contain inorganic sulfide. So these cytochromes and ferridoxins, they contain iron sulfur and this inorganic sulfide, but these rubridoxins, they do not contain the inorganic sulfide. They participate in the electrons transfer in the biological system. The active site of the rubridoxin is the iron, which is coordinated by the sulfur of 4 cysteine residue. So here the iron is not present in the heme group. So it is not a heme protein, but it is a metalloprotein, and here you will find the iron center is surrounded by the four sulfurs of 4 cysteine residues. Okay, and these residues, they are of the proteins. The oxidation state of the central metal ion changes between +2 to +3 during electron transfer. Iron sulfur protein, it acts as an electron carrier, and this rubridoxin, is its oxidized form is reddish colored due to ligand to metal charge transfer. And in the reduced state, the rubridoxin is colorless. So electronic transition actually takes place at the infrared level, and this infrared level is not visible, so these appears colorless. And this is the structure of rubridoxin, and here you can find that this Fe2 center is surrounded by 4s of 4 cysteines from the protein present in the rubridoxin. The third metalloprotein that takes part in the electron transfer is the plastocyanin. And if you see the function of plastocyanin, it is actually a copper containing protein, and this participates in the electron transport process, which is associated with the photosynthetic reactions. So we had already studied the photosystem 1 and photosystem 2 when we had studied about the chlorophyll, chloroplast. These plastocyanins, they are the electron carriers in the photosystem 1 and 2. They act in between these two photosystems. Electron transfer between cytochrome complex present in the photosystem 2 and the entry point of photosystem 1. Okay, so these plastocyanin, they transport electron from photosystem 2 to photosystem 1. In reduced plastocyanin, histidine 87 become protonated, and due to this protonation it can no longer act as a ligand, so the geometry of copper center, it becomes trigonal planar. I will show you later on. First, I will show you the function of plastocyanin, and then we will discuss its geometry. Okay, so this is the function of plastocyanin. This is the photosystem second. We had already studied, and this is the photosystem one. And you have seen that the in the non-cyclic this electron transfer process, the electrons from this photosystem 2, they transfer to some acceptor, and through this to the cytochrome complex, and through this it is transfers to the photosystem 1. So this is the plastocyanin. So this plastocyanin is present here at the entry of photosystem 1. So when electron transfer from cytochrome to plastocyanin and from plastocyanin to photosystem 1. So this is the electron transfer process that involved by the plastocyanin. Now we will see the structure of plastocyanin. So plastocyanin, it has generally trigonal bipyramidal geometry, which is distorted type. Okay, so distorted type trigonal bipyramidal geometry, trigonal pyramidal geometry. It's not bipyramidal, but it's pyramidal, trigonal, distorted trigonal pyramidal geometry. At a trigonal planar position, the active site of the plastocyanin is present. Two nitrogen atoms from different histidines and one sulfur atom from cysteine are present at the trigonal plane, and sulfur from methionine is present on the apex of trigonal pyramidal geometry. I will show you the geometry. The two copper sulfur bond lengths are different and are responsible for the redox potential of the protein. Okay. Again, this copper sulfur bond, it absorbs around 597 nanometer. So plastocyanin appears blue in color. So plastocyanin is blue in color. Now I will show you the geometry. In this geometry, you will find this copper 2 plus is arranged in a distorted trigonal planar geometry, pyramidal geometry. So this S methionine is present at the apex of this trigonal plane. So this is the trigonal plane where you will find two histidine and one cysteine. Sulfur is present as the coordination, and you will find the copper sulfur bond distance at the trigonal plane is 2.13 angstrom, while at the apex it is 2.90 angstrom. So due to this difference, it becomes something distorted trigonal pyramidal geometry. And this difference in the bond length, it causes the redox potential of this particular complex. This is the 3D structure of plastocyanin, where this is the copper ion, and you can find there are two type of histidine, histidine 87 and histidine 37, and one methionine is present here and the other cysteine is present here. So these four ligands, they coordinate with the copper ion. I had told you that in the reduced plastocyanin, this histidine 87, it it get protonated. So it get, if the nitrogen of this histidine 87 get protonated, that it can no longer bind to this copper center, and then the geometry will become trigonal planar. So the reduced form of plastocyanin is trigonal planar because this histidine is not binded to the copper center, but its oxidized form is having the distorted trigonal planar, trigonal pyramidal geometry. Okay, so this histidine in the reduced form, it can protonate it, and then it cannot further bind to the metal center. Now, the second role of these metalloproteins is the light harvesting. So, these metalloproteins, they are also involved in the light harvesting of photosynthetic system. So, these are the metalloproteins that are chlorophyll containing proteins, and these chlorophyll containing proteins are actually the reaction center of the chlorophyll, where the protein complex have chlorophyll A and chlorophyll B. So, we had already studied about the photosystem 1 and photosystem 2. So in the photosystem 1 and photosystem 2, there are reaction centers, and these reaction centers, they are actually the protein complex in which chlorophyll A and chlorophyll B are present, and they acts as the antenna complex, and through this antenna, light can be harvested from the sun. Okay, so this is the very important step of photosynthesis. So chlorophyll, actually it is a metalloprotein which plays an important role is photosynthesis, and it has a chlorine ring. So chlorine ring is different from that of porphyrin ring. It has four polyl ring and one additional ring. So this ring is called as the chlorine ring. It absorbs the solar energy of the photons actually, and then transfer to the reaction center, that is a protein complex having chlorophyll A and chlorophyll B. This chlorophyll contains magnesium as the active site of the chlorine ring, and absorption of photon by the chlorophyll causes an electron to be excited in the singlet and then the triplet state. I had already explained you this mechanism in the lecture that is related to the photosynthesis, the energy transfer processes, the metal complexes and the energy transfer processes. So here I have given only the gist of the how these metalloproteins, they play an important role in the light harvesting system, and during photosynthetic system the chlorophyll is oxidized. Okay, so it can donate electron to some acceptor, and then further it can accept electron from the water molecule which is getting which can form oxygen molecule and eject an electron. So that electron can be accepted by the chlorophyll, and it can transfer further its electron to any acceptor. So during the light harvesting, the magnesium, the chlorophyll, it oxidized and reduced. Okay, it continuously oxidized and reduced. So this is the process of role of the metalloproteins in light harvesting. The third role of metalloproteins is catalysis, and these metalloproteins, they are known as metalloenzymes. So there are various enzymes that I have not taken, maybe taken. So some of the enzymes, metalloenzymes, I had taken in this lecture, which acts as a catalyst and catalyze a number of reactions. So some of the metalloenzymes I have taken, and I had uh, taken the one enzyme is the nitrogenase enzyme, you had already studied. So this nitrogenase enzyme is a metalloprotein where you will find the metal center is the MO. So active site is MO, molybdenum, and this nitrogenase causes nitrogen fixation. And two main features of nitrogenase is the presence of MO as active site and iron sulfur clusters for the transportation of electrons. Okay, so this is the complete reaction of nitrogen fixation, where you will find that one nitrogen molecule will convert into ammonia and one hydrogen molecule, and for this conversion it requires the 16 ATP molecules and 8 electrons. Okay, so here the metalloenzyme, that is nitrogenase. So nitrogenase, that is containing the MO as active site, it is a metalloprotein that catalyzes the reduction of nitrogen to ammonia. Then the other enzyme is superoxide dismutase. So this enzyme we are studying for the first time. So I have given more details about this enzyme. So I have not given the more details about nitrogenase because we had already studied this nitrogenase enzyme. Superoxide dismutase we are studying for the first time. So I have given the details of this enzyme. So this in the biological system, actually the superoxide ions, they are generated as a result of reduction of O2. So if the oxygen reduced in the biological system, it can form the superoxide, and this oxide is a powerful oxidizing agent. So if it is a powerful oxidizing agent, then it can oxidize various kind of reactions in our system, and sometimes it also it generally it kills the harmful microorganisms. So it protects us, but sometimes it in higher amount it damages the cell. So due to the cell damage, the oxide should be killed. So this oxide is killed by the superoxide dismutase enzyme in our biological system. So these are also called as SOD. Superoxide dismutase is an important antioxidant defense in the biological system. So this is the natural antioxidant defense in the biological systems if the superoxide concentration exceeds certain value. So how it works? So how this superoxide dismutase works? It disproportionates this oxide to oxygen and hydrogen peroxide. Okay. So here you will find the oxidation -2 to 0. So oxidation takes place, and this oxide, the superoxide, will oxidize to this O2 and reduce to hydrogen peroxide. Okay, so this is the main functioning of superoxide dismutase. It disproportionate the generated superoxide into oxygen and hydrogen peroxide. How it works? Generally, the metal ion, which shows varying oxidation state, can act as the oxidizing or as a reducing agent. So here you can find this metal center, it get reduced and oxidize the superoxide, and in this reaction this metal ion oxidized and this superoxide ion get reduced. So in this way, due to the varying oxidation state of this metal center, disproportionation of superoxide takes place in presence of superoxide dismutase. There are three major classes of superoxide dismutase. So this depends upon the protein fold, which type of protein is present there, and on the metal ion present. So there are three kind of superoxide dismutase. One is copper zinc SOD, that is present in the eukaryotes cytosol. And here I will show you the structure. The ligands are 6 histidine, 1 aspartate, 1 histidine between two metals. So two metals are present in this superoxide dismutase. One is copper 2 plus and other is zinc. Okay. So generally the copper is tetrahedrally coordinated by histidine. So this is the structure of copper zinc superoxide dismutase. Here you will find 6 histidines, 1, 2, 3, 4, 5. 4, 5 and 6 histidines and 1 aspartate, and 1 histidine is present in between these two metal centers, copper and zinc. So this is copper zinc superoxide dismutase. Now the second type of dismutase is FeSOD, which is present in Escherichia coli and chloroplast, and it is a dimer MnSOD is present in all mitochondria, and here you will find ligands are 3-histidine, 1-aspartate, and a water or hydroxy molecule. Okay, so this is present in all mitochondria. FeSOD may be there or MnSOD. MnSOD is present in all mitochondria. So you can see that Fe, Mn, Fe and Mn or Mn dismutase enzyme is a dimer. So it is a dimer, and one the site active site is, you can see here, iron center is surrounded by 3-histidine, 1-aspartate and 1 H2O or OH. So it is penta coordinated iron center. So this is iron superoxide dismutase. The third kind of this dismutase is NiSOD, that is present in prokaryotes, and it is hexameric in nature. This is the structure of nickel superoxide dismutase, where nickel is the active center of this enzyme, and all these enzymes, they generally disproportionates the superoxide into oxygen and hydrogen peroxide. Now the another enzyme which we will discuss is the carbonic anhydrase, and this carbonic anhydrase role has been already discussed in the transfer of oxygen, that is Bohr's effect. So in the Bohr's effect we had studied about this carbonic anhydrase, and also we had studied in the role of metal ions in biological systems about this carbonic anhydrase. This carbonic anhydrase, it acts on the upon the carbon dioxide. This carbon dioxide is a key metabolite that is generated in the oxidative processes of the biological systems. So carbon dioxide is the metabolite that generates continuously in the biological systems, and this carbon dioxide, it is poorly soluble in the blood, in the aqueous medium, while CO2 is as compared to the CO2, this bicarbonate is it is not dissolved easily in the aqueous medium. This CO2 is remains equilibrium with the bicarbonates, and when it remains in equilibrium with the bicarbonates cannot be easily taken up, but the carbon dioxide can easily be taken up in the form of carbonic acid. So CO2 converts into carbonic acid first in the blood, and that carried out to the lungs by deoxyhemoglobin. You had already studied in the Bohr's effect. So this CO2 will convert into carbonic acid, so that bubbling of carbon dioxide can be suppressed in the blood. So this carbonic acid, it goes to the lungs, and in the lungs this carbonic acid then releases carbon dioxide again. Okay, so in this way it can expelled from the lungs. The dehydration of bicarbonate to carbon dioxide in the lungs is catalyzed by carbonic anhydrase. So this this carbon dioxide to bicarbonate, or this bicarbonate to carbon dioxide, this can be done with the help of carbonic anhydrase enzyme. This interconversion is very important during the oxygen transport. So if this kind of conversion cannot occur, the concentration of H plus ion concentration cannot be changed, which is responsible for the change in the pH of the system, and this change in the pH is responsible for the release of oxygen in the tissue. Okay, so this equilibrium, this kind of reaction, it is necessary part of the biological system for the oxygen transport. So CO2 and bicarbonate, it should be in equilibrium, and this should be maintained in the biological system, and that is done by the carbonic anhydrase enzyme. In the carbonic anhydrase enzyme, zinc 2 plus is the active center, and this active center, it is surrounded by 3-histidine and 1 water molecule or hydroxyl, you can say. So it is 4 coordinated, so it is having the tetrahedral geometry. This zinc center is having the tetrahedral geometry, and when the carbon dioxide bind here, you can see carbon dioxide bind here, so it will convert into carbonate, and this can be shown with the help of a mechanism. This is the mechanism of carbonic anhydrase enzyme, and this is the carbonic anhydrase enzyme where you will find there are three histidines with the zinc second center and one water molecule is there. And this zinc 2 plus is a very important center. Its positive charge increases the acidity of water, so that from this water molecule one H plus can remove easily, and it comes in the form of hydroxyl ion. Okay, and this carbonic anhydrase form, it can easily bind the carbon dioxide, and when it binds the carbon dioxide through this carbon center, then it can be removed. Which can be removed? HCO3. So this is the bicarbonate. So this bicarbonate can be removed by the action of water, so that here water can again joined via this zinc, and then again regenerate the carbonic anhydrase enzyme. So this is the cycle of carbonic anhydrase enzyme, which maintains the equilibrium between CO2 and bicarbonate in the biological system. Another enzyme is the cytochrome P450, which we had already been studied in our one of the lecture. It is a monooxygenase enzyme which oxygenates a wide range of organic substrates. So this is the structure of cytochrome P450, which we had discussed already. Here the iron center is in third oxidation state, and you can see the four coordination through four nitrogen centers of the four pyrrole rings, and one is through the cysteine, and the sixth one is the H2O molecule. So these kind of reactions are shown by the this enzyme cytochrome P450, where you will find the oxygenation of one of the oxygen can inserted in the CH bond. Okay, the organic molecule can be converted into more soluble forms. The other enzymes are peroxidase and catalase, and which we had already studied. The peroxidase enzyme can convert this hydrogen peroxide to water, while the catalase enzyme, it can disproportionate the H2O2 to H2O and O2. We had already discussed their mechanism in detail. These are the structures of peroxidase and catalase, and the only difference in their coordination is there is a histidine in peroxidase that is directly attached to the iron third center, while there is a tyrosine that is attached directly to the Fe3 center in case of catalase. Now the role of this metalloproteins in the storage and transport. The sum of the metalloproteins, they act as oxygen carrier, and among these hemoglobin and myoglobins are the main oxygen carriers, and which we had already studied. I will again show you some differences in between hemoglobin and myoglobin. So both are heme proteins. They are heme proteins because they are having the heme as their active sites, and they can reversibly bind O2. Okay, however their role are different. The HB, the hemoglobin, transport oxygen from lungs to tissue, and this myoglobin take up the oxygen in the tissue from the hemoglobin. Okay, so this myoglobin is present in the tissue while hemoglobin is present in the blood. Okay, then this in the whole part of our body, hemoglobin has four subunits, in which Fe2 is coordinated to the four pyrrole rings and one imidazole nitrogen atom of histidine residue, and sixth coordination is with the oxygen. Hemoglobin has four subunits, so it shows cooperativity effect which facilitates or enhances the transfer of oxygen to the myoglobin. Myoglobin has only one unit of heme and globin unit, so it can only react with one oxygen molecule. These are the structures of hemoglobin and myoglobin. So this is myoglobin, where one porphyrin ring, one heme group, and one globin subunit. So this is a monomer, while this hemoglobin is a tetramer, where you will find 1, 2, 3, 4, 4 heme groups, and 1, 2, 3, 4. They are four subunits of the proteins. So it is a tetramer and it is a monomer. So it can bind only one molecule of oxygen, and it can bind four oxygen molecule to the four heme groups. And these hemoglobin and myoglobin, they participate in the transport of oxygen. This hemoglobin, it takes part in the transportation of oxygen and carbon dioxide, while this myoglobin, it transport the oxygen. It actually it stores the oxygen in the tissue and give up the oxygen whenever it is required by the tissue. Hemerythrin and hemocyanin, they are the two other metalloproteins that participate in the transport of oxygen. This hemerythrin and hemocyanin, they are non-heme proteins. They are metalloproteins, but they are non-heme proteins. They do not contain the heme prosthetic group, and they are found only in invertebrates. Okay, so these are not present in all human system. It is only they are only present in the invertebrates. The iron containing protein in which oxygen is bound it by nuclear iron center. So I am talking about hemerythrin. So in the hemerythrin, iron center is actually the active site, and there are two iron centers here, but it is non-heme. Okay, 8 subunits. It has 8 subunits, and each subunit contain 113 amino acids, and there are two iron centers. And like hemoglobin, it unlike hemoglobin, it does not show cooperative effect. Okay, and there are carboxylic side chains of glutamate, aspartate, and 5 histidine residues at the iron center. This is the structure of hemerythrin, where you will find there are two iron centers that are having the two oxidation state, and there it is present in the high spin state. Okay, and there are 1, 2, 3, 4, and 5 histidines and two carboxylic group of glutamate and aspartate. Okay, so this is the structure of deoxyhemerythrin, and when it reacts with oxygen, then it will form such kind of geometry. And here you will find this Fe center, Fe2 center will oxidize to Fe3 center, this Fe2 center will oxidize to Fe3 center, and both are high spin. And this is oxyhemerythrin, where you will find one oxygen molecule will bind here. In deoxyhemerythrin, Fe2 Fe centers, they have electronic configuration D6, but these two centers, they are coupled antiferromagnetically. So here you will find 4 unpaired electrons, here you will also find the 4 unpaired electrons, but both are having the opposite spins. So if we add the spin of these electrons, the total spin will become equals to 0, because 4 are having plus half spin and 4 are having minus half spin, so that the total spin will become 0. So it is antiferromagnetically coupled, and it is diamagnetic in nature. If we see oxyhemerythrin, then you will find there are two Fe3 plus centers, high spin, so that again you will find 5 unpaired electrons here and 5 unpaired electrons with the another iron center, but both are having the opposite spin, so that the total S will become equals to 0. So again these two centers, they coupled antiferromagnetically. Okay, so they are not showing any signal in the ESR spectra, although they have unpaired electrons. Now if we'll see the hemocyanin, it is a very efficient metalloprotein which transport oxygen in the mollusks. So it transport the oxygen in the mollusks, and in the deoxyhemocyanin, 2 copper are in +1 state, and in oxyhemocyanin, 2 copper are the 1 oxidized to the copper third form. This is the structure of deoxyhemocyanin, where you will find 2 copper centers are there with the +1 oxidation state and 3 histidine around each copper center. And this is colorless and it is diamagnetic in nature. While in case of this complex, this is oxyhemocyanin, after attachment of oxygen in between these 2 copper centers, they oxidize to copper third, and this is blue in color because of ligand to metal charge transfer. So this oxygen oxide will act as a ligand, and the electron will transfer from this oxide to the copper. So it will show LMCT type spectra, and it is diamagnetic in nature. So in this way, the hemocyanin, they can carry oxygen in the mollusks. Now, myoglobin can act as a storehouse of oxygen in the tissues. It accepts oxygen from the hemoglobin and store that oxygen in the tissues. Now, in conclusion, we can say that the metalloproteins, there are two parts in the metalloproteins. One is metal and another is the protein. Metal may be any kind of transition metal ion, so it is different from heme protein. But heme proteins, they are under the category of metalloproteins, where heme is the prosthetic group and iron is present as the active center in the heme proteins. While in case of other metalloproteins, heme may present or may not be present. Okay, so if we see in the gist, if we see the different functions of metalloproteins, then we will find that cytochrome, rubridoxin, and plastocyanin, they are important metalloproteins in electron transfer, and the metal center present in them are Fe, and copper. Then the other kind of metalloproteins are chlorophyll containing proteins, which are present in the reaction centers of the photosystem 1 and 2. So these chlorophyll containing proteins, they are having magnesium in their active sites, and they act as light harvesting systems. Then the metalloproteins that works for oxygen transport, they are hemoglobin, myoglobin, hemerythrin, and hemocyanin, and these are the metal centers present in these metalloproteins. Myoglobin is the metalloprotein that has Fe2 plus center. In its active site, it can store oxygen. While these are the different enzymes: superoxide dismutase, nitrogenase, catalase, peroxidase, carbonic anhydrase. So these are the different enzymes which are having copper, zinc, iron, nickel, molybdenum, iron, and zinc. So these acts as the catalysts in the different catalytic processes in the biological systems. So this is all about the functions of the metalloproteins. Thank you very much.